I am happy to introduce to you all Cameron Moore. Cameron was a student who graduated in 2020, like when everything kind of fell apart. So his last semester here was a very interesting one. He's originally from Clinton, Tennessee, and he was a first generation college student. So that's something some of you may associate with. He was also an S-cubed, which obviously all of you are, or most of you are probably an S-cubed. He was a STEM success tutor. He was an MC ambassador. He was an SI leader for intro cell bio, and he was my TA for genetics and for non-majors microbiology. So I made him suffer through that. And he in 2020 graduated with his BS in biology and a minor in chemistry, and went on into a PhD program in the lab of Dr. Heidi Goodrich. While he was here, he did several internships. He did some research here at MC, but also at OR et al. And then he did one at University of Georgia. And so he got lots of experience that led to him being able to go right into that PhD program. So without further ado.
Thank you. All right, so thank you all for coming. I did not expect such a turnout at 5 o'clock in the afternoon. So today I'm going to be speaking about some of my PhD work at the University of Tennessee in Advisor's Lab, with the title of the talk being, "What is my bacterium eating?" And so this is maybe a little bit more of an untraditional approach to talking about science. It's more of a problem-solving type presentation about how I had to sort of find a way when there was no way in my PhD. And before I get started, I just want to kind of reiterate some of the things Dr. Brigati just said about myself. So, and how I got to microbiology, because I know you're all sort of interested in pursuing some science career and curious how you can get there. So, like she said, I was a student here at Maryville College in 2020, and I was an SU. Here are some pictures from me when I was in the program of acting on the flat water sort of experience, as well as my first day. The other picture is me sort of on my last day at Maryville College that I spent during an undergraduate, where my parents came and gave me a senior photography thing, since I couldn't actually have an in-person type graduation. These are also some fun pictures of me trying to get into the science building to get my microbial cultures I was working on as a TA who lived and left in there. And I just kind of want to speak to the SQ program, our biology program as a whole, has been really profoundly helpful in putting me to where I am today. I don't think I would be a PhD student had I went anywhere else or been a part of a different program. So you all are very blessed and to be in such an awesome program. So like she said, I am a biology, or I was a biology major when I was here. And while I was sort of in college, I didn't really know what I wanted to do science-wise. Like I knew I liked biology, but I wasn't really certain which path to choose. I mean, you've got so many: ecology, genetics, biochem, like there's just so much to pick from. So when I was a junior, I took microbiology with Dr. Brigati. And one of the projects you do in this class is identifying unknown organisms. And so this is the very like auger plate that I struck in intro microbiology that allowed me to go, I really want to do this for the rest of my life. And so what you're looking at is just E. coli struck on a plate that allows it to turn this really brilliant green color. And I thought that's so fascinating that microbes can take simple dyes that they have in sort of an agar medium and turn it into something really beautiful. And so that's what ultimately skyrocketed me into wanting to be a microbiologist. Now, to get there, I had to have a few sort of internship experiences to also soul search for what science I wanted to do. And so I spent a summer working at Oak Ridge in a toxicology group, like after my freshman year. I also worked with Miss Garino, who's the physics professor for those of you who may not have heard yet, working on an eclipse project, so the total solar eclipse of 2017 that came through Maryville. I was on that team of student researchers who took part in that experience. And I also worked at Oak Ridge again in a metabolomics group that they worked on plants. I learned very quickly. I did want to work on plants. And then my final experience was in synthetic and molecular microbiology at the University of Georgia. And so this was a research experience for undergraduates where students go and do research somewhere else for a summer. I know you've had a lot of talks about that this semester thus far, so I'm going to not super talk about how I got it, but if you are interested, please ask questions about any of these experiences at the end of my talk. But while I was at UGA, I started working in the lab of Dr. Eric Stab and with his graduate student, Macy Coppinger. And so they work in a bioluminescent bacterium called Vibrio fischeri. If you've not heard of this organism, don't worry, you'll learn about it in biochem too. But this organism is bioluminescent and it does really interesting chemistry to allow that to happen. But what I was most interested in, it was its association with a squid host. So this is a marine system where you've got a bacterium and a squid living together in symbiosis. And before I continue on, I want to introduce you to the wonderful, marvelous field of microbial symbiosis. So every one of you in this room, and all of these little cartoon animals and plants that I've got on the screen, all have something in common. You all have a very complex and interesting and unique microbiome. And this is sort of the consortium of unicellular, small, microscopic organisms that live in you and on you at any given point in time. So symbiosis is broadly defined as the living together of unlike organisms. And this was a term that was first coined by Anton de Berry, the founder of the field of symbiosis research. And in microbial symbiosis, we often use a lot of nomenclature that the average audience wouldn't understand. So I'll kind of give you an intro to some of those terms that we'll use. For example, the word host may be something you've heard of, but not super familiar. In microbial symbiosis, this refers to the larger partner in the symbiosis. So we are the host. The bugs I showed you in the last picture, the tree, the dog, even little worms in the soil, which I'll talk about later, they are the host organisms in all these situations. The other kind of word you'll hear is symbiote. These are the microbial partner, the smaller organism in the interaction. And so microbes can vary. If you're unfamiliar, there's vast amounts of microbes on our planet. There are bacteria, viruses, archaea, fungi, etc. I'm going to focus most of my talk today on bacteria because that's what I know most about. But if you're interested in anything beyond bacteria, please pursue microbiology. There's something there for everybody. So we define symbioses really in a very scale-like manner. There's all sorts of them, and this is just sort of a short summary of what there could be. As a microbiologist, I'm really interested in symbioses where the microbial partner is benefiting. So they are surviving, thriving, and they're happy. And I'm interested in how they influence host health, development, and things like that. So that system I told you I worked on earlier is an example of a mutualistic or beneficial host-microbe interaction. In this case, that bioluminescent bacteria, if you can see it through my slide, is able to live inside of this squid host in a very specialized tissue, and it will emit a light that will kind of shine through the squid and protect it from underlying predators in the ocean. And so it's benefiting the squid by protecting it from maybe predatory animals. And the squid provides a house of sorts for the microbe in return. There are other great examples beyond squid-type interactions. For example, in plants, there are rhizobia. These are bacteria that can colonize sort of root tissues of plants, and they can fix nitrogen and provide that to the plant, and in return, the plant can provide food. And there's also other types of symbionts, like sort of terrestrial animals, like honeybees. So for one example, the larvae of honeybees have these little microbial symbionts called Bombella apis that provide antibiotics, if you will, that protect these little larvae from being killed off by insect pathogens. On the other side of this, the honeybees provide food sort of in their comb that's around them that the bacteria can feed on. On the other side is antagonism. This is actually what you're probably most familiar with. Any of us who've had an infection from a bacterium at any point in our life will know what a microbial antagonist looks like. So in the plant world, there are plenty of pathogens. This is fire blight. This is a disease caused by a bacterial pathogen called Erwinia amylovera. It is injected into plant tissues by insects, and it ultimately causes disease in a variety of trees and tree-like fruiting plants. In this image, we have a picture of Listeria monocytogenes, and something you might be more familiar with this if you've taken microbiology. This is a human pathogen that's really interesting. So when it infects a human host, it enters the host cell and is able to take over the host cell machinery to move itself through that cell into other host cells. So it's very interesting. And you can see in this image, all of those little red dots are the bacteria, and all of those little green lines following them, that's your proteins moving these bacteria through your cell. So very scary, but also very cool. Finally, sort of just keeping up the bioluminescence theme, there's also bioluminescent pathogens. One here, the one that's depicted here is Photorhabdus luminescens. It's an insect pathogen, and it just happens to be bioluminescent. We're not sure what relevance it has to the actual biology of the organism quite yet, but we do know it exists and can infect insects. And so the reason I sort of just yelled at you about symbiosis for about 5 minutes is to explain that there's quite a bit of diversity across the board of mutualism and antagonism. And broadly, what people are interested in studying in this field is how do microbes take part in these mutualistic associations and antagonistic ones. But my lab is sort of interested in a much higher level question. And that question is, how does a microbe act both as a mutualist and a pathogen? How can you decide if you're going to benefit one host and harm another? And so the model bacterium I work with is Xenoramus nematopula, pictured here in purple. This is a symbiotic organism by every state of the word. It has to live in association with one or two hosts throughout its life cycle. Xenoramus is a mutualist of a nematode, seen here in this gray wiggle, called Steinernema carpocapsine. And these are soil-dwelling nematodes that have a really interesting biological scheme that I'll talk about in just a moment. The bacteria also are pathogens to insect larvae. So this is just Galeria melanella. You don't have to know what that is. It's just the insect we use in the lab most often. And the sort of life cycle of these organisms are really intertwined by this bacterium. So I hope I can coordinate this video well enough. So here we have our insect larva. And you can imagine this organism living in the soil. And so those nematodes I mentioned earlier, they're parasitic to this insect. So they're going to travel through the soil and find an insect host, such as this Galeria that we have here. And so these organisms, oh, it's going to start over. It's okay. These organisms are going to find the insect and enter through a natural opening, such as the mouth, anus, or the spiracles. So in this claymation, they're going to dissect it. And what you'll see is these little green dots start accumulating. The nematodes eat the dots when they leave. And what happened there is that these dots are the bacterial symbiote. So the nematodes go into the insect, defecate the symbiote into the blood of this insect. Then the bacteria will kill the insect, and this bacteria will begin to degrade the insect tissues and ultimately feed themselves and grow and divide and exist happily in the insect. The nematodes would then go in and they feed on these Xenorabius bacteria. This supports their growth, reproduction, development. And ultimately what happens is that they will return to the soil to find a new insect host once they've pretty much depleted the insect tissues. And so that was a lot. And usually when I talk to my students about this, they're like, what's happening? So what I want you to take from this is that Xenorabius is a mutualist of nematodes. So it benefits their life and their development and reproduction. And that it inhibits or harms insect hosts by stopping their reproduction, development, and so forth. And so my question is, how does Xenorabius decide whether or not it's going to be a mutualist to the nematode or a pathogen to the insect? And so most of my project is actually focused on this question of this adaptation. And I'm really interested in nutrients because there's plenty of evidence to suggest that this bacterium senses and responds to nutrients in its environment. And that ultimately triggers these pathogenic or mutualistic phenotypes that we see in the lab. But when you're asking questions about nutrients, you also kind of have to have a background in microbiology and what bacteria eat. So I'm going to do a brief intro on what bacteria can eat in the natural world. So for those of you who maybe have some background, you'll recognize this molecule. This is glucose. This is something you'll find often table sugar or really anything like that. A lot of carbohydrates have glucose. Glucose is sort of the common molecule that a lot of microbes use to feed on. And this is a really energetically favorable and energetic productive molecule. Microbes can also eat amino acids. If you take intro cell biology, I'm sure you can memorize 20 of them. But these amino acids have a very conserved similar structure. They're smaller than glucose in general, but they all have these unique groups on them. If you're interested in those groups, take organic. But all of these groups confer different properties chemically to these amino acids that biologically have different effects when putting together proteins, because amino acids are the subunits of proteins. Other things that bacteria eat include glycerol. So this is sort of a component of fats like fatty acids. I'm sort of giving you a very generic fatty acid image, but you'll also find it in your life in sort of butter, milk, that sort of thing. And bacteria also have the amazing ability to kind of eat smaller compounds like acetate, or sort of the main product in vinegar. And so all of these compounds can be metabolized by a bacterium and utilized for various functions. And so if you take your intro classes, you'll know that they can all be used to contribute to making energy for the cell, as well as building larger complex biomolecules. Now, humans, we can eat all these things, and we actually would have to eat them in order to survive in our environments. If we don't get one or more of those types of groups of chemicals, we will ultimately die or will suffer nutritionally. Bacteria don't have this problem generally, and so they can take one molecule of glucose and convert it down into smaller molecules, and they can take a bunch of glucose and make all of these different molecules, and they can survive on just one source of carbon alone. They can also do this in reverse. They can take these really tiny molecules and build them back up into glucose, as well as building them up into amino acids and glycerol. And so I know this may seem very simple, but microbes have this incredibly diverse metabolic capacity. They can take really anything that they face in their environment and turn it into something else. And so that's why microbes are really interesting and why we're really interested in nutrition and symbiosis. And so we have plenty of questions like, do amino acids confer any sort of advantage in mutualism, or does glucose help in pathogenicity of the organism? And so in sort of starting my dissertation work, I was really trying to find the project, find the data, find the answer to these questions in my own way, and start building a project from them. And so I had to start growing my organisms in media. So if you're not really familiar with microbiology, I've sort of got some generic slides about media making and how that works. So we can grow microbes in different forms. We can grow them in semi-solid agar plates. It's like Jell-O for bacteria. We also have these broth cultures, and we can grow them in various volumes. And so these can be comprised of many different nutrients, and we often will have different specific names for these type of media. So complex rich media refers to these broth solutions that are made of sort of complex arrays of molecules. Someone basically took a yeast, broke it apart, and said, okay, put that in a tube. Now let's grow a bacteria in it, see if it will grow. And so these are called complex rich media because they're rich in nutrients, but we're not extra sure what's in them, or we have some idea, but it's not well defined. So this is a great media type for trying to ask questions that are unrelated to metabolism, or metabolism may have a minor impact. But if you're interested in understanding the metabolic capacity of a bacterium or how it adapts to specific nutrients, you then have to grow it in a defined nutrient minimal media or something like that. And so in this case, it's something where you're taking a certain amount of glucose, amino acids, glycerol, acetate, and adding it yourself to a water-based solution. And hopefully your microbe will grow, depending on the microbe you work with. So if Dr. Bongiorno was here, she would agree with me. Most microorganisms are unculturable. In fact, if you go out into any of the places on our planet and try to grow microbes, you may have some low efficacy on growing microorganisms. And this is actually, there's this concept that host microbiology people like myself have such an easy life because we can culture our microbes, but oftentimes symbiotic microbes form these metabolic dependencies on their host, or even beyond the metabolism. And so they require nutrients from their host in order to grow outside of it. For example, our bacterium is an oxytroph for nicotinic acid. Now, nicotinic acid is important in central metabolism, but I don't actually want you to focus on that. What I want you to focus on is this word oxytroph, because I'll use it quite a bit in my talk. So oxytroph refers to an organism that does not have or has lost the ability to synthesize certain substances required for growth in a given medium in the lab or even in the environment. And so you have to supplement certain nutrients to your bacterium to make it grow. And then the title of my talk is, what is my bacterium eating? Sort of comes to this issue of, I didn't really know my organism was an oxytroph for anything else, or if it was an oxytroph at all. And today I'm going to kind of talk about how I figured out perhaps it might be, at least under certain conditions. So we can culture the Xenobacterium bacterium in a variety of ways. It's actually really well established. It's probably been studied for the past 40, 50 years. And we've been able to grow it in complex rich media like all of these. So if you take microbiology or if you're taking it currently, you'll probably use this in the lab. So this is a medium that's been well established and used to grow many types of microorganisms. And we know that Xenobacterium can grow in this. We also grow it in something called case amino acid medium. My advisor calls it LB light, it's more crystal light, where it's like the diet version of LB. So this is something that's a little bit more defined. We don't actually know everything that goes in, but we do know for the most part how much amino acids are going in. It's amino acid based. We also have lipid auger. This is a really interesting tool our lab has where we can culture our microbes in sort of, together with their nematode host. So this is the only auger medium we really have that we can grow them together and sort of watch their interactions take place. And that's really interesting and cool for our lab. And then we also, for a long time, had a minimal medium glucose recipe where it was just glucose, the nicotinic acid I mentioned earlier, in sort of a buffered water solution, and it grew our bacterium. So before I actually get into my project, I want to give you a sort of run through of how I do a lot of my experiments in my lab. So what I generally do is I grow my organism up in this complex, rich medium overnight. And then the next morning, I will subculture it or take it from that original collection or that original culture, wash it, and then put it into different vials of medium. And so again, there's various ways we can do these sorts of assays. They're called growth assays, and you can do them in flasks, test tubes, or in 96 well plates. And so if you haven't seen a 96 well plate before, they're those little squares with all those little wells. You can add your microbes, add your media, and you can scan a whole bunch of samples for their growth very quickly and very efficaciously. So the output of this in terms of the data is really you can have three different options. The first is optical density. This is probably the most common method you'll see. This simply measures how much light can extend through your microbial culture, so how much goes in and how much goes out, and that can help you somewhat quantify how many microbes are present. For more fine-tuned measurement, you can look at the colony forming units. So if your microbe can grow on a plate, you can take that from the culture, put it on a plate, and count the number of colonies and say, here's how many colonies per mil are in my culture. And probably the most for sure way of knowing how many cells per mil you've got is doing cell counting. And so all of these kind of take different levels of difficulty. For the purposes of my talk today, I'll be talking about my 96 well plate experiments for the most part, and I'm using optical density as my measure. So the output of these experiments are called growth curves. And so here's a growth curve, or at least what a growth curve will get into look like. On your y-axis, you've got your bacterial population densities, how many bacteria are present, based on your measurements, the time elapsed. And when you start a growth curve, you want all of your populations to be the same size. So if you have one population that's small, one that's big, then one will probably grow a lot faster than the other. So when starting these experiments, you have to have sort of a consistent starting population. Now, when you put a bacterium from one medium into a new medium, you're going to have a lag phase. So this is the time it takes the organism to not only adapt to this condition, but also begin dividing enough to reach exponential phase. So this is the phase of growth where the bacteria grow exponentially, and you can enumerate the growth rate or doubling time based on this curve that you get. Finally, the bacteria will enter something called stationary phase. This is where the number of microbes in your solution will sort of peak. They will no longer get any bigger or smaller in terms of the population, and they'll sort of stay that way for some period of time. Now, I told you all this because all of the data I'm about to show you follows this formula. So if you're unclear, just remember, lag phase, exponential, stationary. Okay? So my advisor is actually not from Tennessee originally. She's from the University of Wisconsin-Madison, and that's where she spent most of her career as a researcher. And so she went to UT 7 years ago to become the Department of Microbiology, and she brought all of her bacteria, her nematodes, her insects, everything, and her lab mates, I guess. And when she got here, she realized that they could no longer grow the Xenarabis bacterium on the glucose mineral media. For some reason, it didn't work. No one quite knew why, and no one really needed to know why. None of the projects in our lab at the time were focused on metabolism and really needed this sort of growth medium to do this. And then I joined the lab. So when I joined the lab, my project was really metabolism focused. I sort of gave you that whole preamble about why metabolism might matter in symbiosis. And for about 6 months, I couldn't get it to grow. I had everything I could think of in the lab. I tried to fix every problem, and nothing worked. And I was sort of getting really frustrated. And so I kind of had to lay out on the table what facts I had. And I only had 2. The first fact was that Xenarabis can grow in this LB-rich complex medium. We don't know what goes in, but we do know it'll grow here. We also know it will not grow on this middle medium glucose, and that's pretty much it. That's all I had to go on. All of the papers in the world were not very helpful to me. So then I asked myself the question, could I grow this Xenarabis nematophila bacterium on a nutrient-rich defined medium? And does such a medium exist? Well, the great news is it does. So the medium I found, I actually just Googled it, so the power of Google prevails here in science, was this easy rich defined medium. And this is a medium that's sort of built very similar to minimal medium glucose, but instead of just adding glucose, you add everything. You add amino acids, and I'll tell you a little bit more about all I add later. But it gained its infamy from the E. coli genome project that was conducted at the University of Wisconsin-Madison. But it wasn't new to this sort of one project. It's actually been around since 1974. It was first published by Neidhart et al. And this is like sort of the big folks who studied E. coli before E. coli was cool. And they published that the culture medium that they can make all sorts, nutrient-rich, nutrient-poor, and everything in between. Now, this medium has a lot of ingredients. pH is pH buffered, so for you chemistry buffs in the room, you're probably excited. It's like, I'll just add it and make sure that it works. Trace metals, this is like zinc, nickel, copper, anything that biological organisms need in very small quantities that will help them survive. Inorganic forms of nitrogen, phosphorus, sulfur, and then also organic forms of carbon and nitrogen in the form of glucose, amino acids, nucleotides, and then also vitamins and vitamin precursors. And you probably were like, why are you telling me this? I don't know. But what I do know is that what I'm giving these bacteria is an all-you-can-eat buffet, so like Thanksgiving, and they can eat anything they want in this medium. And so my hope was that maybe, just maybe, my organism could grow. And so the result of this was it could grow. After 6 months of nothing, it finally grew in the lab. And so what you can see is my growth in the minimal medium glucose is absolutely nothing. And in the LB, you see growth, which is in yellow, and then growth in orange on this easy rich defined medium. And so I hope you're kind of recognizing that lag phase, so that really short period at the beginning, exponential growth, and then sort of entering stationary phase. So this was very exciting. I was like, oh my gosh. I told my advisor, I said, Heidi, I figured it out. She's like, well, you got it to grow. Now you have to figure it out. And so my question was, what components of this medium were actually important for growth? And so I started screening my wild type Xenarabis nematophila for all sorts of dropouts. So I took this easy rich medium and just dropped out various ingredients and made various batches. And so there were like 14 growth curves. I've split them down to 3. So the easy rich medium is sort of growth in the 96 well plate. You see yellow. When I dropped out glucose, you can see that there's a reduced carrying capacity, very similar growth rate, and a little bit of a longer lag phase. And this wasn't unexpected. My hypothesis going into this was that it had something to do with trace metals, because our bacterium has a weird relationship with metals. It's actually a big project in our lab. But then I found this really interesting point of data. When I dropped out all 20 of the essential amino acids, there was no growth whatsoever. And this was completely unexpected. My advisor, all the senior grad students, people in the field were like, this doesn't make any sense here. It's not an oxytrove for these amino acids. And I sort of had to work my way through this process and prove to my world around me that this was true. So I asked the question, well, which of these amino acids are actually important for Xenarabis nematophila to grow? So there are 20 amino acids that are commonly found in biology, and performing an experiment where there's 20 different conditions would be absolutely arduous. And I wanted to find an easier way to screen for these oxytrophs. So my advisor did some reading, and she said, I remember this method that we used early on in my lab's history called oxonography, where you take nutrients, so that are in the same biosynthetic pathway. So if a microbe makes one amino acid, the pathway continues and you can make another amino acid. So you can group them together based on their biosynthetic pathway. So for example, here's the aspartate-derived amino acid pathway. At the top of this is aspartate. You've got lysine, threonine, methionine. All of these amino acids are produced by the same pathway in our bacterium, as well as many other bacteria. And so you take these groups that you've identified, and you can grow your organism on those groups. And any groups where the organism grows, you can sort of match that up and say, okay, it's this nutrient that it requires for growth. And so the setup would be sort of the oxonography table. So if your organism is an oxytroph for adenine, it'll grow on column 1 or in pool 1 and grow on pool 6, because that's what is shared between them, right? So I was looking for like this really beautiful curve where I get two nice growth curves from two different pools that say this amino acid. The result was not so fun. So here's an easy rich to find medium in green. It grows fine. You've got 3 pools that show pool 4, pool 9, pool 8. And what you'll see is that there's some growth. Some of them grow better than others. So pool 4 had the most optimal growth. These other ones didn't grow so well. And it was very inconsistent. Some of my replicates, so when you do experiments, you have lots of replicates. Some of them grew, some of them didn't. And at the time I thought I'd messed up, and I told my advisor, I gotta do this again. Well, I did it again and got very similar data. So I was curious. I was like, well, there's some amino acids shared between these pools. Like there was aspartate and methionine, different things present, but I was like, there's not really a smoking gun here. There's not one amino acid that seems to be the issue. So the amino acids within this group, of that group 4, where you saw the best growth, are these aspartate-derived amino acids I showed you like 5 seconds ago, including aspartate, threonine, lysine, and methionine. And for the rest of my talk, I will likely refer to them as the ADAA, which is just my short-term word for saying this, so I don't have to say it a million times. And what's interesting about this pathway is our lab has dealt with it before. It's been a big part of our research, actually. So a lot of the assays we perform in our lab are colonization assays. We will co-culture the bacteria and the nematodes together with mutants of the bacteria to see if there's ones that can't colonize the nematode or those who can. And xenorabies pneumococcal that have lost the genes that confer the ability to produce threonine and methionine, they can't colonize the nematode as well. In fact, threonine oxytrophy abolishes the ability to colonize the nematode. Methionine, to a lesser extent, has a reduced colonization load. So I know, based on sort of papers, that this organism should be producing these molecules. Like there's evidence in papers, we've seen it in the lab. It has to be able to colonize and produce these compounds. On the other side of this sort of lysine side, I sort of said, okay, we know it can make this. Well, we don't know if it can make lysine. We just kind of make that presumption based on genomics. And I did a sort of bioinformatic approach, which is not my best thing in the world. But what I did was I took the protein sequences of the genes within the lysine biosynthetic pathway and aligned them to an organism that we knew could make all of these compounds. And what I found was that overall, all of these proteins share greater than 60% sequence similarity to the cellular organism. Now, that doesn't always tell you for sure that that's functional, that your proteins are working. But when I looked deeper at these sequences, I saw that the active sites, where the proteins are actually supposed to be functioning in this biological system, were well conserved between both organisms. And that was confusing because I was like, well, there's not really one gene that's kind of the one I can say, that's it, that's what's happening. So I performed another growth curve experiment. And what I did was I took just a glucose mineral medium that I'd made in the lab and added all of these amino acids, whether in single addition, so like aspartate alone, threonine alone, methionine, lysine, or in combination, all of them together as the ADAA, or sort of just aspartate, methionine, threonine. And what I found was that they all supported growth in various ways. So for example, all of the single additions, after about 30 hours, started to grow. Not super great, but they did grow. And this was interesting to me. It's not one amino acid that's rescuing this phenotype. In fact, in addition and altogether, you get this really nice growth on glucose. And I thought, okay, well, if I at least have to add only 4 amino acids, that's not a big deal. But this was really new to my lab. And so I said, okay, what if this oxytrophy is only specific to glucose? There's a lot of colloquial evidence from my graduate student friends who said, well, someone said that xenorabies grows really well on glycerol. And someone was like, it could grow on acetate. And so I did an experiment and I said, okay, does the addition of these aspartate-derived amino acids confer any sort of growth on all these other carbon sources, or can they grow alone? And my findings were pretty interesting. So sort of as a positive control, I grew Xenorabbis on a amino acid only medium. The reason I did this was because we know it grows really well, it likes them a lot, and it grew the best of all of them. We also found that the glycerol, when grown only with glycerol, no amino acids added, it grows amazing. It grew almost the exact same as when glucose was supporting those amino acids. And even acetate, that teeny tiny molecule that I showed you earlier, was a better carbon source than glucose, which is really surprising because most microbes can't eat acetate or can't eat it really well. They don't love to. And you will notice that there is some growth in glucose after about 40 hours. So I have grown this microbe in glass culture tubes. I've grown it in 5 ml culture tubes. It's never grown after a week in those sort of environments. In the 96 well plate, very inconsistently, I will get a phenotype where it grows. This is not common. This is not usual. It just showed up in this one growth curve. So my hypothesis is that there's potentially a mutant that's sort of arising after like 2 days sitting there in 96 well plate. But there's further experiments I need to do to make sure and validate this. So now that I know that this oxytrophy is conditional, it's not specific to just all growth, it's actually only on growth on glucose. I wondered if this was an issue with our strain of Xenorabbis gametophyla. So if you're interested in microbiology at all, one day you're going to join a lab and they're going to say, here is our type strain, the strain we use to conduct all of our experiments, and everyone else uses a different. So there's several in the field of Xenorabbis. There's all of these that I'm going to show you here. You don't have to remember any of them, but they're all considered strains of the same species. They're just distinct subtypes of one another, meaning that they all are roughly the same organism with just a little bit genetic diversity that makes them a little bit more distinct. So they have different phenotypes. And so I was just curious to see if all of them have this phenotype, because the concern was that our wild type Xenorabbis was a mutant. Somehow in the traveling from Wisconsin to Tennessee, our Xenorabbis accrued some random mutation that alters its ability to grow glucose. And it was sort of a way for us to say, okay, let's just see if everyone else has the same problem. So the growth curve is really nice. So I had 4 conditions. I grew all of these strains on glucose and amino acids, so all 20 of them added together. They grew amazing, seen here in black. They also grew amazing with these amino acids alone, and on glucose, every single one of them didn't grow except for one. It's like one that came out of the woodworking for this experiment. And this was not one of the ones I usually work with. But when you added the aspartate-derived amino acids, every single strain improved their growth on glucose, and you can see them. And so what this told us was that this phenotype is conserved among these strains. This is something that could be very specific into the biology of these organisms. And so this was really exciting because this means I found something. So the next question to ask was, is this oxytrophy that I found, this conditional oxytrophy, is it conserved among other Xenorabbis bacteria? So what I haven't shared with you yet is that the Xenorabbis gametophyla is one of many Xenorabbis that exist on the planet and that have been isolated. So here's a co-phylogeny. So this is putting together the Xenorabbis bacteria with their nematode hosts and seeing how similarly phylogenetically they co-evolve. And what you'll notice is there's a lot of Xenorabbis bacteria. I don't know how many actually, but there's a lot of them. And in red, I selected these isolates because of sort of where they fell on this phylogenetic tree. For example, you'll see Xenorabbis gametophyla here. It's sort of the biggest one there. Actually, it's some closely related organisms and some more distantly related organisms. And do you remember earlier when I talked about that bioluminescent like bacterium that infects insects? I also used that one. So this is sort of our outgroup. In this co-phylogeny, it is not a Xenorabbis bacterium. It's a very different group. And it was sort of just as a control to say, it should probably grow, right? And so just to reiterate, these are different nematode-associated insect pathogenic bacteria. They all are sort of differentially related, think like they're cousins. So you've got first cousins, then cousins, sort of thing. And so the growth curves weren't that pretty, so I presented it in a table. And what you'll see is that all of them grew with glucose and amino acids. All of them grew on amino acids alone. All of them grew when glucose was supplemented with those aspartate-derived amino acids. And only 2 of them grew on glucose. The rest of them were not able to grow on glucose, which means that this conditional oxytrophy is conserved among several subgenera of Xenorabbis or subspecies of Xenorabbis. And so this was really interesting, and this will hopefully be a part of my PhD work going forward, because now we've got a good case to say there's some really interesting biology going on in terms of glucose metabolism. And so to kind of wrap up my talk, I want to kind of have some conclusions. So the first is that I hope I convinced you to some level that Xenorabbis is an auxotroph, is aspartate-derived amino acids, when grown in media supplemented with glucose. I also hope that I convinced you that this is not specific, or this is specific to glucose and not specific to other carbon sources like glycerol or acetate, which are utilized by bacteria in a variety of environments. and that this auxotrophy is conserved among both strains of Xenorabbis nematophylla and among other nematode-associated pathogens. And so going forward, I have a few questions I want to ask. So one question is to kind of look at more carbon sources or carbohydrate resources. Glucose, as I said, is sort of the big metabolite of all biology. Like it's what we use almost in every way to do metabolism. So could it be that Xenorabbis has lost the ability to metabolize glucose efficiently, and it sort of just does it as an auxiliary function? I'm also interested in understanding how maybe glucose metabolism or amino acid metabolism may be important during host interactions. And this is a question that usually, people say, don't ask stuff like that, because like glucose metabolism is going to be important. But maybe in this case, it won't be, or if it is important, it's not specific to all of this life cycle that it endures. And finally, my sort of ultimate question is, why do some Xenorabbis grow in glucose while others do not? So those two that grew earlier are very different from the rest of their cousins in this phylogeny that I showed you. One of them only is a pathogen to crickets. And then both of them, the other two, the two that grew on glucose, share a really interesting phenotype. They don't colonize the worm very well. They have a very low colonization load, which could suggest that there's something about glucose metabolism that's not advantageous under certain host microbe interactions. And so with that, I will take any of your questions. Okay. Who wants to go?
Okay, so they'll grow in glycerol.
Mhm.
And then grow in the aspartate derived amino acid.
Mhm.
Glucose plus.
Mhm.
Did you say if you just use aspartate, they'll grow in aspartate?
So I've not done aspartate and find them alone. They don't grow very much. So the amount of aspartate derived, amino acid I'm adding is very low, like 0.0008 molar of any of these. So it's a very low amount of them that sort of supplements growth. So because I did a growth curve, just adding what I would usually add, because the question that we had was maybe they're just eating those and not the glucose, but they don't reach a very high carrying capacity. It's like an OD of 1 at best, or 0.1 at best. Just like,
Well, I'm just thinking that whatever is going on, must be in the early steps of glycolysis, because glycerol will about midway through, and so if that can be used as an efficient food source, but glucose can't, it must be something must be going on. At least if maybe there's multiple problems.
I think it might be a glucose transporter. So I proposed this to my advisor the other day. I said maybe it's the transporter, because it wouldn't make sense to sort of say the first half of glycolysis doesn't work, like it's sort of the central carbon metabolism. So it might be that the advantage of getting glucose in the environment isn't really that advantageous, because so what I didn't mention, sort of the other level of dimension, is that Xenorabbis does sort of primarily pathogenize these insects, but it is not the only organism there. In general, multiple Xenorabbis can infect the same insect. You can have competitors from different whole clades of microbes. And so I did an experiment that was sort of it was unrelated, but I was growing E. coli and Xenorabbis at the same time in amino acids, and Xenorabbis was way faster than E. coli on the amino acids, which is kind of unexpected. E. coli also is known to really like eating amino acids first and glucose. So I think that it might be a competitive advantage in an environment where all microbes are looking for maybe glucose. I'm going to eat the amino acids instead. And we also have a good body of research to say that the nematodes are rich in amino acids, at least in that sort of the colonization area. Because in order to colonize this, plenty of auxotrophs from different amino acids could colonize pretty well. There was only really 2 that were the problem. So I think that it might be that they just adapted to a lifestyle where amino acids are their primary source of nutrients, and maybe cholesterol as well. Quite sure. Good question. Yes.
Really good talk. I think you should go into teaching.
Thank you.
Those 2 strains that you found that have the same phenotype, are they closely related to each other?
So no, Graphiniae is down here, and Nexi is all the way up there. So they're not super
So do
related.
you know why that? I mean, I
No idea. This is sort of very recent discovery. I was like, oh, you both grow? I don't know why. The Annexi organisms, that's the one that I said it's like a specialized pathogen for just crickets. And it has a very interesting biology, but it's also going to be pretty finicky in the lab. Like we can't do genetics in it. And these Xenorabbis bacteria in general are pretty easily genetically modified, except that one. So there's like something about the next that's very distinct, and no one's been able to crack the research code of that. Graphene is interesting because it's our newest sort of addition to our array of Xenarabis. We actually have an isolate that's up and coming. It can be genetically modified, and its nematode host can be modified. So this is something that our field has suffered from a little bit, is that none of these nematodes really have that genetic modification like C. elegans has, because they're not, most of them I don't think are hermaphroditic, but the host of X. graphene, at least when we've isolated, is hermaphroditic. So hope is that, I think, to start building on that and look at host by microsets, which could help with these questions maybe. Any questions? We can also ask my grad school.
I'm gonna ask a non-science question for the
Okay.
benefit of the students. If you can talk about the adjustment of moving from undergrad to grad school.
Yeah, so I had a different adjustment maybe than most because I started during COVID. But I think one of the biggest things for me coming from here to UT is just the size of the school I was kind of going to. And I actually didn't really get knocked over by the size difference until classes were in person again after COVID ended, if you will. There are so many people at UT. I can see how many people get lost in such a large school. So that was a really big kind of change for me, was getting to go from, oh, hi, Dr. Gibson, nice to see you again for the 15th time today, versus the millions of people I see every day at UT. And I also think it was different going from a very didactic and like a classroom type education style to then all of a sudden a very practical in the lab, reading papers, more apprenticeship style of learning how to do science. So you go from being like textbook, taking notes, doing tests, and then going right into I'm a science in the lab, trying to do stuff. So that was it. That was an interesting change for me.
I'm just curious, have you done your first preliminary exams yet?
Yep, I actually just got done this summer.
Maybe you can tell them about that.
Yes, so part of the PhD process, I think at all schools, probably for at least biological sciences, is you take a qualifying or preliminary exam. And so at UT in microbiology specifically, this is an exam where you write a grant. So like one day if you decide to become a scientist that runs their own lab, you write a grant to get money. So you write sort of a fake one, if you will, for this test that is about your research project. So like I wrote mine on a very different project at the beginning. It's since changed because of this, but I wrote my grant and you submit it to a committee of professors. So for example, if Dr. Brigati was my actual major advisor for my project, then I'd have Dr. Ray, Dr. Gibson, maybe Dr. Swan on my committee. We could then they would assess your work and say, okay, did he do a good job at writing this? Did he communicate this? Does he have enough data? Is it rationale, or is it a good rationale and premise? And they sort of assess you on that. And it's a 3-hour long test where you stand up here just like I'm doing, and you present it just like I did. But while you're talking, the professors interject and they're like, hey, why did you say that? That's wrong. Let me tell you. And you have to sort of go back and forth. If that's a good way to answer. It's certainly a very trying part of graduate school. I think it's been the hardest part for me thus far, but once you're done with it, you're sort of in a calm. You're like, okay, I'm here and I'm here to stay. Yeah. See stress faces now that I've said this.
Could you speak to like your schedule now as
Yeah.
a graduate student?
So my schedule has changed dramatically. So, I guess going here, you have class at like 8, 10, 11, then you have lab like 2 to 5 every day or something like that. As a graduate student, your schedule is a little bit more open. So the classes you take, if you when you take them, are very participation based. You show up, you listen to the speaker, you go back to the lab, or if you do take a class that's very sort of test intensive, that normal didactic class, they don't take up a lot of your schedule. It's like maybe one class a semester like that. So I often find myself with like large batches of time. And the reason you kind of have that time is to be in the lab doing research. The only thing that really, I think, changes dramatically, other than maybe taking fewer classes, is you start teaching. At least in part, some graduate students teach to get their funding to stay in graduate school. And so teaching takes up a large amount of your time, especially depending on the class that you're teaching. So I teach an advanced micro class. So I teach 2 days a week, 3 hours every day, or each of those days. And so that can be a little bit of a time suck for my research, but I like it. I find it kind of rewarding to be said. That is, your schedule will change. You'll become the teacher. That's terrifying. Any questions? My students are quiet too, so I'm sitting here all day long.
Do we have any other questions for Dan?
If you're an introvert, you can ask me stuff after too.
All right, so let's give Cameron another round of applause.